Good afternoon, everyone, and welcome to Grand Rounds. My name's Tom Farden, and I'm the chair and organizer of Grand Rounds. Now, it's funny how some of these Grand Round topics end up coming to me. Josh, who's our speaker today, emailed me a few months ago saying that he perhaps had a talk he could present in the new year about phage therapy, but it was a bit tentative. And then I got on a plane to go to a conference at the end of November, and just randomly I was allocated a seat next to John Narge, one of the vascular surgeons, who sat down next to me and launched into this conversation about phage therapy. And did I know this guy called Josh Jones, who was really interested in phage therapy, and would it make a really good Grand Round? So this got me approaching me from two directions. Well, this is definitely something we need to look at. And Josh got back in touch with me, and today is the day. So I know very little about bacteriophage therapy. In the world of respiratory medicine, there is a fairly famous case of phage therapy being used in mycobacterial disease, tuberculosis. But I understand that phage therapy has wide-reaching possibilities and wide-reaching therapeutic targets, but it's I think Josh will allow me to say it's a fairly controversial area, and he's pushing on with phage therapy here in Dundee to become a centre where we can push the research on and start to develop a clinical service. So that's about all I know about phage therapy. So I'm really interested in hearing from Josh today. If you have any questions, please put them in the chat at the chat box. We can ask real life questions at the end. I think Josh is talking about maybe 40 minutes and time for questions at the end. So Josh, thank you very much for taking the time to come and speak to us, and looking forward to your talk.
Perfect. Well, thank you, Tom, for that introduction. So my name is Josh, and I'm leading the new clinical phage lab here in NHS Tayside, and more on that a little later on. But in the next 40 minutes or so, I'll hopefully introduce you a little more to... all right. I'll introduce you a little more to phage therapy. And sorry, I'm just going to stop the video. I don't really see my ugly mug as I present. So there are two parts to this talk. So the first part, I'm going to introduce you to phage therapy. We'll go through a little bit of the background about what phage are, how phage therapy works, what's the evidence for it, and look at some of the caveats as well. And in the second part of the talk, towards the end, I'll introduce you to the new clinical phage lab here in Tayside. So what are phages or bacteriophages? They are literally, the word bacteriophage means bacteria eater. So as a virologist, I sort of subscribe to the idea that everything has a virus, and just as we have viruses that cause us problems, so do bacteria and almost all manner of other organisms. In fact, there are even viruses, but we won't get into that. So viruses that infect and kill bacteria are bacteriophages, and they're known as phages typically for short. So phages were discovered serendipitously in 1915 by this British gentleman, Frederick Twort. He was doing some vacciniovirus research, putting a few bacterial agar plates together, and noticed these rather strange clearings on his agar plates. Those, of course, were phages and what we call plaques. Each of these would be the clearance of bacterial cells due to the killing action of initially just one phage particle that has replicated and caused this clearance in this bacterial lawn. So he spotted these, and then unfortunately his research was interrupted by World War I. He went off to fight, and when he came back, he didn't pick up his research. 2 years later, phages were also independently discovered by this chap, Felix Durel. So he was a Frenchman. He was a bit of an outsider to the establishment. He was an autodidactic scientist. He actually initially was interested in distilleries, and he'd been off in Mexico looking at agave plants and tequila distillation. And the problem was that they had lots of issues with locusts eating their agave plants. So he had the idea of, well, if he could find a sick locust and isolate something from that sick locus, could he use that to control locusts? So a sort of biological control, if you like. He then moved on and applied his idea to chickens, but this time he was looking for something in chicken feces that could cure bacterial infection. And at this stage, all they really knew about phages was that if you had a culture of bacteria, it was cloudy. If you filtered fecal matter, in this case, through a sufficiently small filter, that filtrate could be added to a culture of bacteria, and that would turn that bacterial culture from cloudy with lots of bacteria in it to clear with lyse bacteria. And that was all they knew, that there was something filterable that could cause a bacterial culture to clarify. So he then moved on from chickens to patients, and it seemed a natural progression. And in 1919, he oversaw the treatment of the first patient in Paris, which was actually a child with dysentery. And that child was treated using filtered feces from a convalescent Shigella patient. So if you're interested, by the way, in the history, this book here has, I'm sure there are more up-to-date books, but this has quite a nice overview of that. It's a very curious history. So phages then look like this, and this is an electron micrograph of a phage on the left and a schematic on the right. The genetic material is encoded in the head of the viral particle here. Effectively, what happens is phages will bind the surface of the tight bacteria through these tail fibers. This sheath then compresses as these tail fibers, sort of, it's hard to illustrate on Zoom, but they sort of lift up and the head pushes down, a little bit like a hypodermic needle. And there are specialised enzymes at this base plate which will allow a hole to be made in the bacteria and the genetic material inserted. Phages have two replication cycles, broadly speaking. So the simple replication cycle, which we'll start with, is the lytic replication cycle. So in this case, a phage has bound to a target bacterium, insert its genetic material. We've got a transcription, translation of that, production of new viral particles, and lysis of that bacterial cell. And that will just repeat. And obviously, you might have a small number of, perhaps even a single phage or a small number of phages binding initially, producing thousands of more phage particles, and so that amplifies the number of phages. Those are the types of phages that we're interested in for phage therapy, lytic, strictly obligate lytic phages. I'll just mention it briefly for completeness. The other replication cycle is known as the lysogenic cycle. In this case, some phages will, the genetic material of some phage will integrate into the bacterial DNA. The reason temperate phages do this, and these are actually, I'm going to go out on a limb and say these are actually more common than lytic phages, particularly in our microbiome. The reason they do this is because they effectively link their own survival to the survival of the bacteria. So the phage genome here is replicated as the bacteria replicates. So some phages that exist in this form encode elements that promote bacterial survival. So it's not uncommon to find phage toxins on here, like cholera toxin, diphtheria toxin. Those are phage encoded toxins, but also antimicrobial resistance genes may be on there as well. When you've got a quiescence, a cell with a temperate phage in it, if that cell undergoes environmental stress, and there are a variety of stressors that might meet that criteria, that phage can excise itself from the genome. It senses it's on an evolutionary sinking ship, becomes viral particles again, and can then reinfect new cells. So that's a very sort of whirlwind tour of phage biology. So we are always in contact with phages, and we've co-evolved in constant contact with phages. There's an estimated 10 to the 31 phages on the planet, and this is then a constant pool of viral genetic material that's turning over. And phages have been found in all sorts of sort of strange places that were previously thought to be sterile. So particularly CSF, for example, blood. Phages are also a little understood, but probably very important part of our microbiome, and I think there's something like 10 to the 9 phage per gram of fecal matter. In terms of the number of viral particles, in an average 70 kilo male, it's been estimated that could be something like 4 times 10 to the 14 viral particles. Now, most of those are probably going to be phages. So we are absolutely packed with and constantly exposed to phages. So in terms of the history of phage therapy, we've touched on that a little bit, and we've said that the first child treated, or the first patient treated, was this child, Robert Kay, in the Sick Kids Hospital in Paris in 1919. And that really heralded a golden era for phage therapy. And phage therapy was used widely and enthusiastically across the West and elsewhere. There's a nice paper in the Lancet Infectious Diseases about phage therapy in South America at this time as well. And it was very successful, such that it even made the headline of the New York Times in 1925. People were very, very excited about this prospect. You could buy phage products over the counter, and there were large-scale field trials of the sort we just wouldn't conceive today, where they effectively took two villages and that were suffering cholera epidemics and dumped phage into the water of one but not the other, and watched to see what happened, and with varying degrees of success, but obviously they were pretty poorly controlled. So phage therapy had a real sort of heyday here. And what happened is that antibiotics then, well, there were a variety of factors. Largely antibiotics came along, and at the time antibiotics were much easier to make, to market and to use, and you didn't need to know the patient's bacteria or the sensitivity of the patient's bacteria for antibiotics at the time. Whereas with phages, because they're so specific about the bacteria they infect, you really need to do what at the time was called phage typing to check you've got a phage that matches that bacteria. Otherwise you wouldn't see an effect. But there were also manufacturing challenges with phages as well. So in the West, we had this kind of golden era, and then we really saw a sharp drop off in favor of antibiotics. Meanwhile, in the geopolitical East, so we're talking Eastern Europe and Russia, they established the Eli Arbor Institute, which exists in Tbilisi in Georgia, the former USSR, and still has one of the world's best, if not the world's best, phage collection. There was mass use in the Red Army and civil society. They took a few hits during the Cold War and the purges, but they are still using that. You can still buy that over the counter. It's still used in hospitals, and I've got some in my fridge from Georgia here. So it's still a fundamental part. It's not novel in the geopolitical East. Over here in the West, though, we've now got perhaps not just an awareness of AMR, but also perhaps of the significance of antibiotic tolerance has really driven renewed interest in phage therapy. So let's briefly look at how phage therapy works. So this is just to illustrate that if you have a collection or a library of phages, you can use that in two ways. You can make pre-formulated phage cocktails, which you can then use either preferably based on screening, but certainly you could give these empirically. This is an example of a Georgian cocktail, not too dissimilar from the one that's in my fridge. The alternative way, and quite a powerful way to use phages, is personalized medicine. And so if you have a collection of phages, you can take a patient's bacteria, you can test that bacteria against your collection of phages, choose the optimal phages for that, and provide a personalized formulation. Now, in the long term, the sort of long-term vision for phage, certainly in the UK, is effectively to replicate what they do in Georgia, where we have pre-formulated cocktails to meet most clinical need, and those would be licensed therapeutics, much like antibiotics, and dealt with and overseen by clinical microbiology. But also then to cater for a minority of patients who would need personalised phage therapy, because either they're not covered by this or because they've got bacteria that have generated resistance to this. So that's the sort of the long-term view. So in terms of the advantages of phage therapy, well, obviously we've got bacterial killing independent of antibiotic resistance. I'll talk a little bit later on about this in more depth, but there's no notable side effects from phage therapy. And that's not a surprise when we consider how intimately in contact we are with phages and how we've evolved with phages. Also, they're suitable for patients, of course, immune deficiency, because they're not a pathogen. Because they're so specific, they're considered to have very little impact on commensal flora. Phages have this curious idea of being auto-dosing. So if there are lots of bacteria at your site of infection, you end up with lots of phages because you've got lots of phage replication. But as soon as there are no bacterial hosts left, the phages are either excreted or rapidly destroyed by the immune system, because phages have no immune evasion capability. Phages also have antibiofilm activity, so this sets them apart from antibiotics. Some phages have specialised enzymes to help break down that biofilm matrix, but phages can also infect the sort of dormant cells or the quiescent cells that exist in biofilms, and they don't then immediately replicate. Phages need to be replicating in a cell, but they sit there like a time bomb, and when that bacteria in that biofilm starts replicating again, that allows that phage to replicate, and that will not be good news for that bacteria. There's also increasing evidence, both in vitro evidence and clinical evidence, as we'll touch on later on, that phage and antibiotics can act synergistically. In some cases, they can act not antagonistically, but if you use an antibiotic and you've killed all your bacteria, or you've prevented all your bacteria from replicating with a bacteriostatic agent, then you obviously won't have any effect from phage therapy. But there is increasing evidence that these two evolutionary angles can really put bacteria in quite a sticky situation, and compromising for one can lead to increased susceptibility against the other. Relative to antibiotics, phages are pretty cheap and fast to produce, and we've talked already about the potential for personalised medicine. I've included a few references on the right here, which may be of interest. This one at the top was one I wrote with a consultant in GD&C, and it contains a reasonably good general discussion of these advantages and caveats of phage therapy. So moving on to the caveats, we'll start with the fact that perhaps obvious, but you need to have lytic phages for your target bacteria. Now that's sometimes straightforward, sometimes it's not. It's not always easy for every bacterial species to find lytic phages. So Tom mentioned a case from, I think Tom was touching on the Great Ormond Street case, which was published in 2019, where an engineered mycobacterial phage was used to treat a patient. The reason that was engineered is because a lot of mycobacterial phages are temperate phages, as we saw earlier. They have that cycle where they integrate into the mycobacterial DNA. You can engineer out those genes that are responsible for that and make them obligate lytic. So that is where it can be a little more challenging. Also, if you've got a polymicrobial infection, you need phages for all the pathogens that are responsible for that infection. So again, that can, certainly at this very early stage, that can be a little more complicated. Phages generally have quite a narrow host range, and that is, the specificity has benefits, as we've seen, but it also makes it tricky. But you can overcome that by using phage cocktails. I touched on bacterial resistance to phage. So bacteria can and do become resistant to phage. If we think about it from an evolutionary point of view, bacteria and phages are in a constant evolutionary arms race. Now, phages are smaller, they've got a smaller genome, they can evolve faster, but bacteria will develop resistance to phages. Not always during phage therapy, but if that is encountered, the way around that is simply to switch phages, because you've got such a diversity of phages available. The immune response to phages doesn't generally get in the way, particularly for acute treatment of acute infections or relatively short courses of phage. There have been some patients with mycobacterial infections in which we've seen neutralizing immune responses. Now, again, the action to take there is to switch the phage if that's possible. These immune responses don't always interfere with the activity of the phage. Sometimes, if they're sufficiently neutralizing, then they can, and then you have to switch your phage. In the long run, there are two ways to mitigate the activity of the immune system. One is to train your phage. So that is sounds like the title of a good film, but that is actually in vitro directed evolution. So replicating your phage in the conditions in which you would like it to be used, i.e. something close to the human body. Or, as I understand, I think has been the case in some of the Georgian cocktails, actually to simply passage your phage through people, and sequentially passaging your phage through people then yields a phage at the end, which is less readily cleared by the immune system. And we've also, at this early stage of phage therapy, we've got limited access to repeated courses, which is a bit of a practical obstacle. And of course, phage therapy can't solve an underlying predisposition to infection. And perhaps most crucially, actually phages need to come into contact with the bacteria. So that sounds simple, but This means there's challenges for intracellular bacteria, particularly mycobacterium. If you've got a patient with a systemic infection, lots of extracellular organisms, those might be easily cleared by phage therapy, but intracellular reservoirs will almost certainly remain, and that infection may come back. So you can use phage here to knock down an episode, but whether you'll get full clearance is very unlikely. Equally, if you've got pockets of sequestered bacteria, for example, in epithelial layers in urinary tract infections, they can be tricky, and you may not get them all. So I just wanted to say a word about the clinical phage diagnostics here. So lots of people, including us, are still relying on classical microbiological techniques. So we'll place out the bug here in the lab and look at it in liquid broth and try and see how well that phage kills that bacteria. More generously funded labs have advanced clinical diagnostics, and what they can do is use real-time high throughput or provide real-time high throughput data on bacterial growth in the presence of, they're using 96-well plates, so they can do loads of combinations. So for example, here we've got bacterial growth only. Then we've got these 4 colored lines are how the bacterial growth is affected by each of these individual phages. And we can see each of them knock the bacterial growth down slightly. But when you combine those 4 phages, you've got a very potent cocktail that keeps that bacterial growth down. So these platforms are, I would like one of these platforms. These platforms, I think, are very powerful because you can also look at real-time phage antibiotic synergy in this with different combinations of phage and antibiotics. Some people also think then that in the future, we might not even have a lab, and one day this will all just be AI-based diagnostics and we'll print out the right phages. But personally, I think I would always be reassured by some form of functional assay. So in terms of the global state of phage therapy, so phage therapy is being used successfully, albeit sporadically. It's certainly in the Western world, but worldwide phage therapy is being used in these sorts of places. And as I've said, in Eastern Europe, Georgia, Poland, Russia, it's much more accepted. There's also not only civilian interest in this, but military interest from the perspective of biodefense and also the treatment of soldiers with resistant infections. So in terms of sort of Western phage centers and networks, there are a few that have popped up over recent years. So there's a big movement in Australia with a phage biobank and a sort of a large clinical trial that the protocol has just been published for. There's an established phage centre in Belgium at the Queen Astrid Military Hospital, and these are the guys that we'll collaborate with probably mainly for what we're doing here. They've been using phage on a compassionate basis for the last 10 years or so, and their interest is they're a military hospital, and they came into this looking at soldiers with resistant burn wound infections. Also got phage centres set up in Israel and a few in the States, and this company has just received a sort of $10.2 million contract from the US military to develop this further. So we've been relatively slow to get going on this in the UK, and that's largely because of the lack of our own phage manufacturing. Currently, all UK cases rely on imported phage, but also a misconception that existing medicines regulations haven't catered for phage just because phages are so different from chemotherapeutics. And that's actually not the case. Existing regulations are sufficient for phage therapy, and phages can be used in the UK as an unlicensed medicine in cases where licensed alternatives, i.e. antibiotics, are not meeting a patient's clinical needs. And that guidance is set out in MHRA Guidance Note 14, and it's then up to the local NHS unlicensed medicines policy to oversee that. In Tayside, I think that's called the non-formulary request policy. So far in the UK, we've had 2 patients who've received lengthy courses of antimicrobial phage. I think Tom was alluding to this case earlier. And we've had 10 patients treated in Scotland. I oversaw this in Lothian in GG&C, and this is currently in submission. I've also done a quick sort of straw poll of consultants in Scotland, and it seems there's real clinical appetite for phage therapy for patients whose infections antibiotics are not necessarily meeting the needs of. So the sorts of scenarios in which phage could be potentially appropriate would be chronic infections that on paper may be susceptible to antibiotics perhaps, but in practice are not, perhaps due to antibiotic tolerance or biofilms. Patients where there's acute risk despite appropriate antibiotic treatment, completely resistant infections, which are actually pretty rare, but would certainly be a candidate. And then other circumstances that preclude the use of appropriate antibiotics or mean that an alternative medical option is needed, perhaps high-risk surgical cases. So let's just look at the evidence for the safety and efficacy of phage therapy. So there's a substantial body of clinical data, much of which comes from around 2,400 patients treated since the year 2000. And these cover a wide range of infection types. And these were patients whose infections were generally untreatable or refractory to antibiotic therapy. And I've just included a few efficacy rates, indication-specific efficacy rates from systematic reviews down here. Now, the caveat here is that this is observational data, and so we can say, well, the failures might be underreported. Perhaps this isn't appropriate. But I think what is reassuring about this body of observational data is how many different contexts and how many different groups and clinicians all seems to get very similar outcomes. So I think we should probably keep that in mind. Now, the other issue here is that this number is actually a substantial underestimate of the true global size of phage data. And if you remember that West versus East split in the history of phage therapy, actually it turns out there's an awful lot of data that we can't access. There have been some attempts. So this dossier here was a translation of a fairly sizable translation of Georgian literature. And that was published in 2009, funded by a UK defence threat reduction project. But there is a lot of literature that we can't look at, and it is also the case that in these contexts, it's probably unlikely that where phage therapy is not seen as novel, that they even bother publishing in sort of routine uses of phage therapy. So I mentioned that phage has few adverse effects, and I apologise. I'm not expecting anybody to read this slide. Apologise for the busyness of it, but I just wanted to show you these are all the modern reports of adverse effects that I'm aware of for phage therapy. I won't go through them in detail, but none of these adverse effects have been definitively caused by the phages themselves. The most curious of these is the 5 instances of raised LFTs with IV anti-staph phage. So none of these patients, and these reports were, I think, all in 2021. That's this report with 4 patients and this report at the end here. None of these patients actually were reported to have symptoms, and the LFTs normalized either when phages were stopped or when phages were simply continued at a lower dose. It's not entirely clear why that's happened, but in contrast, that there are also lots of other reports of IV anti-staph phage use without deranged LFTs. So what's going on in those cases, it's not clear. It's unlikely that this is being caused by the phages themselves, but it could point either to some sort of contamination in there or impurity in their phage, in those particular phage preparations, or it could point to the role of the liver in helping clear large amounts of foreign protein that's just been injected into the blood. So other sources of adverse effects have generally reflected either been related to the route of administration, or, as I've mentioned, impurities in phage preparations, or debris from bacterial lysis due to phage killing. So actually, for example, if you had a patient with E. coli 0157 and you gave them a phage that killed all their E. coli, it wouldn't be good news for the patient, because the mass lysis of 0157 would release an awful lot of Shiga toxin. So actually, a substantial lysis of bacterial cells can itself cause adverse effects. So moving on to the clinical trial data. So I've said that there's a lot of, or a sizable body of observational clinical data that's very reassuring. There have been 13 clinical or safety trials since the year 2000, and by a variety of routes of administration. And all of these have shown phages to be safe, but the efficacy signals have been mixed. So only 2 of the 7 modern trials that have looked at safety and efficacy have actually shown evidence of efficacy. So there is a glaring discrepancy between this substantial body of clinical case data, which looks very compelling, and 7 clinical trials, which actually look a little bit shaky. And this can be explained because phages just simply aren't that straightforward to use. You've got to get the right phage to the right place at the right time to treat bacterial infections containing sufficient bacterial cells to allow phage replication. So if you get one little bit of that constellation wrong, then you won't get efficacy. And the bottom line is then that and I've just written a review on this, the bottom line is that 5 of these 7 trials have simply got 1 or more of those elements wrong. And the 2 trials that have actually shown efficacy have got all of those elements right. So there's a bit more on that in that paper up there. So phage therapy also poses some challenges for clinical trials. So the specificity of phages means that you've got to have excellent microbiological diagnostics, and of course we do here in Tayside, to make sure that you've got the right bug. And although there's such a diversity of phages, that can also be a problem for clinical trials. It's absolutely impossible to trial every possibly clinically relevant phage or combination of phages. You just can't trial that. There's also the slight complication that almost all the patients will be receiving concurrent antibiotic therapy. So there's a challenge of just teasing out quite what's happening and which effects are due to the phage and which are due to antibiotics, or is it due to the synergy. Arguably, this is a little easier if you're working in patients who've got documented antibiotic refractory infections. Then there comes a really big issue, which is development and supply. And aside from the usual disincentives that pharmaceutical companies face in terms of developing new antibiotics, let alone antimicrobials, phages are not protectable. We're talking about naturally occurring phages, and so these are not patentable. These are discoveries, but they're also very easily copied. If we find a phage here, there's nothing to stop somebody else buying that and then pretending that they've discovered it themselves, because how could we prove otherwise? So those sort of caveats have put development, or financially put development of phages off. There's also then limited access to GMP phage therapy, or where there is access, it's extremely expensive. So there's a trial for Pseudomonas infections in CF patients in the States at the moment, and they're paying about £750,000 for phages for about 50 patients. So that is an obstacle, and certainly trialing a dynamic phage library model is not going to be possible without, well, financially, let's put it that way. And finally, there are a couple of other challenges that phage preps regularly need updating. So if you do a trial of a cocktail, you get a cocktail through a trial, that cocktail is going to need periodic review to keep pace with bacterial evolution. And also then trials of single phages are actually pretty not useless, relatively limited value, because you're always likely to want to use phages in combination, although the phage we will have it here in Tayside is an exception to that rule. So I just want to finish this section by summarising 2 key papers from last year. So first, there's a systematic review on the left, which documents over 2,000 clinical cases that showed excellent efficacy, and concluded that the phage was well tolerated. And when they actually looked at clinical trials that were included in this review, they found that there were more adverse events among the control patients in those trials than there were among phage patients. The other paper, significant paper that came out last year, is this one from the American Antibiotic Resistance Leadership Group. And they published these considerations for the use of phage therapy and suggested that there's sufficient evidence for phage to be considered in antibiotic refractory infections, and that phage is generally safe to administer with adverse effects rarely reported. And you might be interested to know that Health Improvement Scotland have also been reviewing the value of phage therapy for antibiotic refractory infections, and their report is due out soon. So I thought I would include a case, but I'm looking at the time. Tom, how are we doing? Shall I move on?
I think you're fine, don't worry. Fine,
okay. So this was quite a high profile case, and we'll just go through it briefly. So this was in 2017. And I'll just introduce the characters here. So we've got the patient, they're all, you'll spot the connection, they're all at the University of San Diego. The patient here actually was a professor of psychiatry who was married to a dean of global health sciences. And we'll go through the case in a moment, but suffice to say this was a successful case, and the physician in this case and the patient's wife now run this phage centre in the States called IPATH. And they've also, Stephanie Strathley has also written a book as well, which I'm sure you can find on Amazon or all good bookshops. So this was a 68-year-old male who had necrotizing pancreatitis complicated by multi-drug resistant Acinetobacter pseudocyst. he was not in a good state. Antibiotic therapy had not yielded success. They screened his isolate against 98 phage from the US Navy collection. Now, that was a collection that the US Navy had been building up for sort of 10 years or so. And that's been, I think, bought now by a private company, as is the way with these things. And they, within 18 hours of getting this guy's isolate, they had a personalised phage cocktail for him. So he was still on meropenem, but they then they added in this phage cocktail, and we'll look at what happened in a moment. He was started straight away with phage via percutaneous catheters. So they went for a local administration approach. And you can see here, this is one of the plots that I alluded to earlier with their advanced diagnostics, that this is one of these phage cocktails, and how well that kept the bacterial isolate down. So over the next 36 hours, this patient's condition didn't really change particularly. He was still unresponsive, intubated, and his renal and liver failure wasn't improving particularly. So they thought about it, and they considered that the infection might actually be more widespread than where they were reaching with their local administration. So they started IV phage. And I'll just draw your attention to this plot on the right then, which comes from that paper. So they've given this patient IV phage. This is the PFU per mil, or active phage particles per mil, in this patient's serum, 5 minutes before IV phage, 5 minutes after, and so on. So you can see how quickly IV phage is destroyed by or is lost in serum. So by the time we get to 6 hours later, there's none, and there's almost an imperceptible signal at 3 hours, and barely. This is in phage terms, this is barely anything by an hour. In fact, even not a lot by 30 minutes. So the issue here is that there is a little how the value of IV phage. Well, we'll see in a minute that this patient. For this patient, IV phage seems to make the difference. But the point of including this is to illustrate that IV phage, and a question remains over how much phage administered intravenously actually gets to the site of infection. And there is a hole there, I think, for in terms of data. But as we can see, and as you can see from other cases that are published, IV phage seems to work. But I suspect an awful lot of phage is lost almost immediately due to complement and other innate immune defenses, because phages do not have any way of evading that. So for this patient, he starts on IV phage. Day 2, he comes out of his coma, he's convalescent for the first time in weeks. Day 4, actually they pick up for the first time an isolate of this Acinetobacter that's susceptible now to minocycline, so they start him on that. But by day 8, there's bad news, and we've got a phage-resistant isolate. So what they actually do in this case is they go out to raw sewage, and they find a new phage. So they take this patient's bacteria, they filter raw sewage, they challenge that filtrate against this patient's bacteria, and they isolate phages that kill that patient's bacteria, and they add that into the phage cocktail. And that then kept that bacteria, that Acinetobacter isolate down. So the patient then had prolonged phage therapy. We've got a total of 18 weeks of intraperitoneal and 18 weeks of IV. He continually improved once he started phage therapy, and he had no adverse effects. So that was a case that in 2017 really sparked an awful lot of enthusiasm in phage, particularly in the States. And there was also a case where we started to see a bit of phage antibiotic synergy in practice. So the original phage cocktail, actually they found, if you look in this paper, it lost its lytic activity. So that original phage cocktail they used on its own couldn't hold that bacterial culture down, but it could hold it down in combination with minocycline. And again, minocycline on its own couldn't hold that down, but could with this cocktail. But they also interestingly observed morphological changes in this isolate. So on the left here, and the image isn't wonderful, but on the left you can just about make out this kind of grey halo, this capsule on these Acinetobacter organisms here. These are then sequential isolates, and by the end of I can't remember how long the time scale is here, but in their final isolate here, you can see this capsule is they saw loss of capsule and increased extra polymeric substance. And the idea here is that actually, if the phage is binding to the capsule, the bacteria is down-regulating that, and it's increasing this kind of extracellular substance to try and keep the phage at bay from it, and in doing so, perhaps makes itself a little more susceptible to minocycline. So that was quite a nice example. So that is, I'm just looking at time, I'm just about keeping to time. So that was a whirlwind tour through phage therapy and an introduction to that. I'll just now move to the second part of the talk and introduce you to the Tayside Clinical Phage Lab. So this is a one-year project we're running to the end of this year, funded by the Tayside Health Fund, and what we're doing is supporting access to phage therapy, and it's run by myself here in level 5 with support from Emily, who's a trainee biomedical scientist working with me. So we can support access to phages for appropriate patients, although I should stress that we're a scientific and logistical service. We don't make any clinical decisions, and I'll come on to the governance in a moment. We will have anti-staph phage on site, and that will be mainly active against Staph aureus, but it's an unusually broad spectrum phage. So I've spent a while saying how specific phages are. This phage is the exception to the rule, and it does have activity as well against some other Staph species. We have a small budget as well to source phages from international centres for other types of bacteria. And although access to Staph phage, once it arrives, should be pretty quick, it may take a lot longer depending on the source for access to other phages. It's very hard to estimate our maximum patient capacity, as each patient will probably need different amounts of phage, but it could be something in the region of sort of 60 Staph patients. But in practice, if we reach half that number, I would be surprised. In terms of non-Staph phage, we're really limited by the goodwill of the international centres, many of whom will supply phage to us for free, but also the high costs of isolate export and phage import. And then just to add that any patients treated will be patients who should have been treated in Tayside regardless of the availability of phage. So this is a very busy slide, and please don't try and read it. I apologise for it now, but it's just to summarise the governance process for phage requests. So first, you would need to engage with whoever your infectious disease or microbiology colleagues in the way that you normally would. And the first step is to have a brief sort of go, no go discussion with them to scope out any alternative licensed options. And this isn't the final decision, but it's an initial filter. Phage requests can then come to our shared email, which you probably just about read there, and it's at the end of the talk. But any requests we've received that have, for whatever reason, not been through that initial filter will then be reviewed at this stage again by a consultant microbiologist. Any requests that then make it that far through and come to me, I'll evaluate the phage options, and we ask then that you take verbal consent from the patient for screening of their isolate against the phage. If it's a Staph isolate, we'll do the lab diagnostics here. If it's non-Staph, we'll try and source suitable phages, and we'll have to send that isolate abroad. Once we've got a suitable phage, then the usual MDT with input from ID Micro, the phage lab and antimicrobial pharmacy will develop a treatment plan and submit that through the normal non-formulary request policy pathway. And if that request is then approved, a written consent form must be obtained from the patient for using the phage-specific consent form, and you'll also need a phage-specific prescription and administration chart. So that is a very brief overview of the governance. I'm just looking at the time. I'm very grateful to the AMG for their input on that, and they very kindly uploaded that to their website. So there is a link. If you go on, I don't know if you can see it at the top. This is the Antimicrobial website. On the left-hand side in blue, it is Bacteriophage, and the link is at the bottom there. I've also, if you click that link, it will take you through to here, and the staff information leaflet, which contains much more comprehensive information than I would have time to give you this afternoon, is on there. There's also a patient information leaflet and a consent form. I've asked for this to be available on Referral Finder as well, but I don't think it's up yet. So I think I've just about kept to time, perhaps 5 minutes over. I apologise. I'll stop there, and I'm happy to take any questions. Thank you.
Thanks very much, Josh. That's really, really fascinating stuff, and really intriguing that it's been around for such a long time, yet something I was taught zero about in medical school, for example, and not something that really we get taught about at all. Obviously, I'm aware of the Great Ormond Street case, as you alluded to, with dramatic results, and the case that you went through again. That chap, pretty sick, ICU, end of the line, and in the end, 18 weeks worth of phage therapy plus the minocycline got him better.
Yes, and I think the cases in Great Ormond Street, I think was 32, no, not 32 weeks. I think Great Ormond Street was the best part, between 6 months to a year. The second case at Great Ormond Street was, I think, whatever 1.7 years is in months.
Long time. And it's very interesting that our own innate immune system does actually clear these things very quickly. As you were saying, you know, complement probably is a big part of getting rid of these viruses, that within hours you're pouring a huge amount of phage which is into the system, and it's only a small amount that's left. Very interesting stuff. So for patient access, you've given us a nice clear view here. This is for patients who've got resistant organisms, have got persisting infections, got sort of end of the line, what else is left sort of stuff.
Yeah, so as I said, we're using, I'll just scroll back. So these are the sorts of scenarios that it may be appropriate to use phage in, and that would be a discussion between yourself and your ID or your usual ID micro contact. But yes, phage will be used as an adjunct into antibiotics in these cases. I probably should have made that clear, that if you have a patient in one of these scenarios, they will still, or probably almost certainly, unless they're contraindicated, still be on their antibiotics, and we will simply add phage into the mix. That's
really good. Thanks very much. Any role for prophylaxis?
So
I know the answer to that, but
the answer is yes in the future, and certainly if we look at the, I'm just going to go back up that Georgian document, or that translation of Georgian, I'm on the wrong slide, that translation of Georgian literature that I showed you before has a huge chapter on the prophylactic use of phage in that context. In our context, at the moment, the short answer is no. Unlicensed use is limited to those scenarios that we just saw on that slide. Maybe one day, yes, but not now.
I mean, the patient you think of is the recurrent UTI patient who's on repeated doses of OPAT and increasingly resistant ESBLs, etc. And you just wonder if that. I'm just spitballing here, but you know, through a catheter, intra bladder installation of phage, who knows? That
would be a discussion between you and your ID micro contact.
So there's a question here from Mark Spears. Unless he wants to turn his microphone on, I'll just read it. As I'm at the age where I feel I make statements rather than ask questions, I feel compelled to state the following. Oh, crikey. If we lived in a sensible country, the NHS would control the manufacturing of off-patent medicines critical to patient management, which would then allow provision of phage to patients who could benefit in addition. But of course, we don't. Mark is one of my chest consultant colleagues based in Perth.
I agree. You know, this I would like to see this. You know, the only obstacle to this happening is money, basically. And the frustration is that, you know, the private sector doesn't want to invest in it because it's not particularly attractive because of the reasons that I mentioned. My initial thought was that it would fit very well as a public sector service. We'll see what Health Improvement Scotland have to say. There are other boards interested in using this at the moment. Lothian GNC have obviously used it before. GNC are very keen on using this as well, but we will not progress beyond ad hoc cases and limited availability until we've got our own phages. And I think it would fit very well in some sort of, or as some sort of standalone NHS national sort of specialised service. But whether the funding is available for that or not is an entirely different question.
Mark just wants to wish you best of luck getting the government to take this on. It will flourish once taken on by this sort of system. So there's
yes, it will. Thank you.
Are there any other questions from the audience? Anyone got a hand up or just put a question in the chat? Josh, that's been very interesting, really very interesting, and an excellent presentation of the science and the clinical, the clinical application of real bench to bedside stuff. Petros Paskanos has just said we will hopefully soon try it on the first orthopedic patient for a prosthetic joint infection, and clearly that with your staff phage, that is clearly a really target patient population for whom we we're further on now. We were 20 years ago with OPAT, ticaplatin, etc. But this clearly, we'll see how this goes. What I'd like to do is invite you back maybe towards the autumn of this year, so you can feedback on how many patients have come through the service.
Absolutely, yeah, I'd love to.
Let us keep us in touch. Let us know what's going on.
Yep.
And really, yeah, great to hear from you. Thanks so much.
Fantastic. Well, thank you very much for having me. And if anybody wants to email me or with follow-up questions or drop by, we're on level 5. Do come and say hello. And thank you very much for inviting me.
No problem at all. And next week at Grand Rounds, CAMS here, Dr. Fiona Moore is presenting on an as yet to be announced title. So I can't give you any sneak preview at all, but I will send you an email early next week and let you know what the talk's going to be about. Grand Rounds is here every Thursday in term time, 1 p.m. The link is the same every week on Zoom. So copy the link into your diary, just click on it, we'll be here. If you've missed any of these Grand Rounds, you can check them out on our YouTube channel. Just go to YouTube and search Dundee Grand Rounds, and we're the first thing to click on. Please do subscribe, click the wee bell, leave a comment, click the thumbs up. It makes me smile, if nothing else. And thanks so much. Enjoy the rest of your day. Have a good weekend. See you next week.